Hello, my name is Bayarbaatar Amgalan from NCBI NH. Uh, in this talk, I will be presenting one of our recent studies on understanding the etiology of mutagenic process in cancer. The title of my talk is Influence Network Model Uncovers a Relationship Between Biological Processes and Mutational Signatures. Cancer um, somatic mutations occurs in all cells of body throughout the life. They are frequently caused by endogenous or exogenous factors such as exposure to UV radiation or to certain chemicals. Most of research in cancer genome have been focused on identifying cancer-driving mutations which confer growth advantage to cancer cells. In addition to these driver mutations, cells also accumulate a large number of mutations, seemingly no direct role in carcinogenous paths, which might be resulted from the cumulative consequence of DNA damage and deficiency of DNA maintenance mechanisms. The patterns of somatic mutations in cancer genome can be computationally examined as cocktail party problem, in which there are multiple microphones recording the conversations, and the goal is to use these recordings from the microphones to separate distinct conversations. Similarly, we have cancer genome acting as microphones and recording the activity of the different mutational process. This mutagenic process leaves different patterns of somatic mutations called mutation signatures. The different cancer genomes can be subjected to distinct levels of exposures to such mutagenic process. In other words, a unique combination of such mutational process represent the patient-specific mutational loads in cancer genomes. Currently, more than 100 mutational signatures have been identified across human cancers, and the etiology of some mutational signatures have been identified, but however, exact role of many mutation signatures are not understood yet. Now the question that comes up is how to identify the etiology of those signatures. In this study, we developed the network-based method for identifying direct relationship between mutational signatures and molecular pathways. We assume that the identification of direct neighbors of mutational process would help us to understand the role of mutational signatures, and particularly, we expect that the uncovering up and downstream activities of mutational process would provide insights into mutagenic process. And we asked how these mutational signatures are interact with the other process during cancer progression, and also do they communicate with each other. So to this end, we analyzed an integrated data set of gene expression and exposure to mutational signatures. So as described above, the signature exposures are the activity of mutation signatures in cancer genome. The patient-specific activity of a mutational mutational process is represented as the exposure of the corresponding mutation signature. So for genes in the network, their activities are represented by the gene expression, and the goal is to identify genes or molecular pathways contributing to the exposure of mutational signatures or influenced by the mutational process. Here is our workflow of our method. So first, matched samples of gene expression and exposure of mutation signatures are giving us input, and genes and signatures are considered as nodes in the network. And our method consists of two main steps. In the, um, in the first step, the sparse estimation of a full order partial correlation is obtained by uh, sparse partial correlation method. Especially the SPCS is an existing feature selection method to select direct neighbors of variables with their contribution weights. And here the W is a non-symmetric matrix uh, uh, which is a partial correlation matrix, and its non-zero elements represent the connectivity relationship between nodes in the network. In the second step, step for each pair of nodes having partial correlation effect on each other, the partial Higher moment based strategy is uh, used to decide direction of dependency, and finally, a weighted directed network represents the dependency flow over genes and signatures in the network. So as I mentioned, the second step of our approach is proposal to resolve the directions for bidirected edges in partial correlation network. And the problem is how to identify dependency direction in the presence of potential compounding effects that comes from the other variables. So idea is simple. First, remove the compounding effects from the focused pair of pairs coming from the other variables. Then under compounder-free assumption, we use a Higher moment measurement to assign the direction of the dependency. Here is a simulation example to explain the idea of using a partial Higher moments to resolve directionality. So we have two correlated variables, X and Y. We also consider two compounders, Z and U, influencing in X and Y with some partial correlation effects. And our goal is to decide the direction of dependency between X and Y under the potential effects due to the presence of compounding factor and Z and U. So this in this study showed the applicability of partial correlations to remove effects from observed data. So inspired by these findings, we used our partial correlation coefficients to remove the compounding effects from the observed activity of X and Y. And once the compounders have been removed, the corresponding residuals are assumed to follow bivariate distribution. Then we use a theoretical results that Higher moment statistics are applicable to indicate the influence directions between two dependent variables from the purely observational data. And we found that the affected variable is closer to normality than the influence factor under compounder-free assumption. So we implemented these findings to decide the directionality as shown in the simulation example. So the direction is, our decision is, the direction is from Y to X, X to Y. So here is an example showing how in experimentally identified direction is confirmed with the proposed partial Higher moment based strategy. And in figure A, the two Higher moments measurements provide contradictory directions based on the observed gene expression data. After removing compounding effects from the expression data, the residuals are likely more correlated, and partial Higher moments correctly identified the experimentally validated directions based on the shape of residual distributions. So we compared the performance of our method with the several existing causal inference methods using simulation data. And for simulation of the direct sparse network, we implemented the data simulation scheme introduced in this Lengam paper. And according to the simulation study, our method performed better than the most of the method. Only the GenNet method was competitive with our method. In biological systems, the assumption that all compounding effects are present due to only observed activities might be oversimplification. So it's important to evaluate the method on real data. And to test this, we applied our approach to TCGA, breast cancer, and lung cancer data, and performed a similar comparison with existing methods. We used protein DNA interactions as reference, and it is the ChIP-seq experimental data, and genes and signatures. Gene signature networks include genes between each between genes and between signatures and between gene and signatures. However, we have only able to test the predicted directions between genes using the validation set. So as shown in this figure, the GenSignet performed better than the compared methods in this data set. So for the analysis on breast cancer and lung cancer data, we consider genes involved in go-immune response and DNA metabolic process, and the mutational signatures, we included only the signatures known to be operative in the cancer type. So from the analysis on cancer dataset, many relationships uncovered by the GenSignet method are consistent with our current knowledge about the mutational signatures. For example, we found a strong positive influence from HRD mutations and to double-strand break signatures. And in addition to our result, includes no negative impacts of BRCA1 gene expression on double-strand break signature, which is also consistent with the role of BRCA in homologous recombination deficiency. And in addition to the consistency with the existing knowledge, we identify several interactions suggesting link between biological pathways and mutational signatures. For example, we found an interaction between apoptotic hypermutation and activity of regulatory T cell in breast cancer, and the relationship between apoptotic mutations and change in DNA confirmation in lung cancer could also be interesting to look into. And our result also suggests a possible link between signature 8 and near pathway in breast cancer. So as the brief introduction of contributors in this project, Teresa is a group leader, This work was completely supervised by herself, and Yua and Damian worked on this project, and we thank our group for helpful comments. Thank you for listening. I will take question now.
All right, thanks for a great talk. So I'll get the questions started again. I'm curious about whether or not you've benchmarked any of the method relative to perturb-seq data or some type of high throughput perturbation data.
So what data?
Any type of high throughut perturbation data.
Yeah, I think only the data availability is, I mean, for example, the mutation data for the perturbation might not be available. That's the only issue too. We don't use that.
Okay, thanks.